What I'd like to do now is turn the floor over to Jeff Messer. So Jeff, back from GSK, he's the director of analytics. He's been there for quite a number of years. He has a ton of experience under his belt in just practicing basic DEL, but he's going to talk to us today about machine learning. And I'm sure a number of us, I in particular, am looking forward to this talk. So take it away, Jeff.
Great. Well, thank you, Gwen. Just doing a quick sound check. Can everyone hear me all right? Give me a thumbs up. I see you nodding. Great. So, so I thank the organizers for the invitation and the sort of opportunity to try to give a reasonable and rational talk in 10 minutes. So I guess we'll see how we do. Okay. So title of the talk today is DEL plus PDE3A. So that's a safety target that we have at GSK. And I think the key thing here is that I'm going to touch on and focus on is the interpretable ML portion of the talk. Sort of coming late in the sort of cycle here for a 2-day conference. I don't have to really give any DEL background, so I can just kind of give a real quick, computational viewpoint on what DEL is. So, DEL is Katie pulls the library pool out of the freezer and hands it to the biology group that goes and generates data for us computational scientists. That data is essentially an accounting of things that bind the target of interest, in this case, PDE3A, right? And those would get classified as positives or ones. And then there are things that don't bind, or they bind the affinity matrix, and those would be classified as sort of zeros. And so in sort of the machine learning talk, we're going to have a sort of binary model, right? And that, sort of binary model then sort of processes all the data and makes recommendations for things that you might want to buy or acquire. And this is an Amazon warehouse to sort of illustrate that. And Amazon goes ahead and sends you compounds without the DNA attached. And those are going to be kind of the things that will be further sort of looked at with assays, binding, sort of biochemical, cell-based, that sort of thing. All right. So a little bit of background, again, briefly. So inhibition of PDE3A is associated with cardiotox. We run an assay every week to sort of support our lead op campaigns to make sure that we're not picking up any unanticipated off-target tox. We have, for all of those kind of off-target things, we have internal QSAR models. This particular one has somewhat limited domain. We have about 2,000 unique to GSK sort of data points, 50-50, in terms of sort of greater than micromolar potency or not. And the goal here is to basically to come up with new opportunities to make a better QSAR model. So we want to identify new chemotypes for PDE3A to improve the domain of applicability of the QSAR model. So basically the model's only as good as the data you feed it to train on. It can only go so far in terms of interpolating or extrapolating. So the goal here is to feed it more information. Now, one can imagine this would also be useful in a sort of, a lead discovery campaign as well, right? In terms of identifying potential chemical matter that might hit your target of interest. So, jumping right into it, we built a PDE3A model on DEL data, and the standard sort of machine learning way of evaluating the quality of your model is to hold out data from the training and see how the model does on that holdout data, right? And so I won't go into many details on the model build other than, and that's just based on time, I used an unfolded Morgan fingerprint. Again, a lot of detail here, but I hope this, makes sense to some of our sort of chem informaticians and folks that are sort of interested in the technical end of things. To featureize the molecules, and that fed into an XGBoost learning sort of model. And our training set size was about 1 million compounds, and we had about 279,000 columns, right? So it's a pretty big spreadsheet. It's a pretty sparse data set, and we used actually a sparse representation, which is sort of what drove us to XGBoost, because it does a great job of sparse data. And again, only about 5% of the molecules in the training set were positive, right? So those were things that were sort of got flagged as binding the target. Most of the things in the training set were background binders or just things that are, in the library and weren't selected. Again, the supervisor for that training was a binary classifier. And I also sort of bolted on an uncertainty model and domain of applicability sort of calibration. There's a reference down here from Bob Sheridan, who's been publishing in this domain for a long time, but we don't really have time to kind of go into that other than I'll sort of show, the value that that brings, right? And so, in terms of summarizing, the performance of a model, I find it most useful to just look at what's called a confusion matrix, where you basically take the predictions as columns. And so each column here is the prediction, and then the actual ground truth is the rows. And you just basically sum up, the things that, occur in one of the four possible sort of outcomes, right? Where, in this case here, these are things that were negative and the model predicts them to be negative. For example, these were things that were positive and the model predicts them to be positive, right? And in the case of a binary prediction, something is positive when the probability of it being a 1 is greater than 0.5. Okay? And so, without going into too much details here, you basically have two calculations on this matrix that you need to pay attention to. The first is sensitivity. So, it's basically of the positives in my sample, what fraction of them does my model get right? And then the other thing is the precision, or, I like to call this sort of the virtual screening hit rate. And this is of the things I've flagged as being positive or potentially interesting, to sort of pursue, what percentage of those actually turn out to be positive, right? And so, there's always a trade-off between sensitivity and precision. And then just to sort of illustrate the utility of the domain of applicability model, I just want to sort of show the confusion matrix again after applying the calibration for domain of applicability. And, I want to emphasize, I've now switched how I'm defining something that's a positive. I've basically moved towards what's called adjusted odds ratio, where the probability of being a positive is sort of normalized to the probability of being a negative that's been corrected for the uncertainty estimate that you've gotten out of the data. And, basically, if you're more likely, even with the uncertainty, to be a positive, I'm now calling you a positive. And when you sort of apply that heuristic to the same data, right, that has been used, sort of held out without, being included in any of the training or any of the uncertainty estimates, you do see a decent bump in the sensitivity and, a really nice bump in the precision. And, it's the precision that I think is going to, sort of play forward in sort of the most useful way. So anytime you build, so I want to emphasize, when I talk about having built a model, it's always on Dell data for this talk. And then all the predictions I'm showing you from here on out will be on data for compounds that do not have DNA attached. And so the first place I went to was, a bank of compounds, Chembl, where one can easily find active compounds for many targets. And there's a lot of literature actives. In fact, there's 297 literature actives in Chembl. So thank you, Chembl, for that, because it makes it a lot easier to put a presentation together to share sort of publicly available chemical structures. And then the second thing, you need is decoys in the terms that are used in the virtual screening community, or sort of things that are almost certainly negative. And in an ideal situation, you want those to be property matched to the actives, so that you're not just following up, your machine learning models and just learning like molecular weight makes something more likely to be active, right? And so the Schreikert lab out of UCSF has really made some great tools available. And I just went to their sort of tldr.docking.org site that sort of uses a process they called, and I don't know, apologies if there's anyone familiar with this, I don't know if it's called DUDS or DUDES, sort of process to sort of create a set of decoys. And so I did that. I used those sort of two publicly available tools, and before I sort of give you sort of how well the DEL model did at predicting the publicly available data, I just want to sort of point out that, sort of if you look at molecular weight for the actives pulled from Chembl, that's sort of this panel here. You can see the average molecular weight, nice, sort of crisp 350. Right, and if you use the, if you look at the average molecular weight of VLT training data, I don't think anyone's going to be surprised to see, we're a little more bulky, out here with the mean at, 430, 435. So there is, an 87 Dalton difference between those means. It's definitely a right shift, right? And so, one of the first things you'd sort of worry about would be, is this going to sort of inhibit my model's performance? So the kind of the next thing you look at is how much overlap is there from a sort of chemical similarity perspective between, the training data and the sort of external validation data. And so that's what this sort of panel here demonstrates, that actually, there's very little chemical similarity based on sort of simple Tanimoto, coefficient, right? And so, where there's nothing actually, within 0.6. And, if there's only 87 Daltons difference, I'd say that robustly, the structures from the DEL that we're hitting PDE3A are very, they don't have much similarity with the structures in the validation set. And in fact, if you just use chemical similarity as a really sort of simple, kind of old school virtual ligand-based virtual screen, you see essentially no enrichment. So I don't have time to go into the ROC curve background, but basically you create a ROC curve to compare stuff. And if the diagonal, the line basically follows the diagonal, there's no enrichment. So now moving on to the machine learning predictions, since we've looked at a baseline and we've sort of compared chemical similarity for the DEL model predicting the Chembl sitting in sort of the set of decoys provided by the Shoykit Labs sort of server, we can break that down into there's 297 actives, and there's a very large set of decoys that are property matched. I also did a clustering. It's just simple Butina clustering on that data set to understand, sort of the secondary measure, which would be not just how well are we doing at picking out single compounds as being active, but, how well are we doing at picking out chemotypes, right? And I just, so we'll get to that. So when we take the predictions from the machine learning model based on the DEL data and ask it to tell me who's active for the Chembl data, we can see that we have, a reasonable or a relatively low compared to the DEL holdout set sensitivity, right? So the domain of knowledge for the DEL is different than the domain of sort of knowledge in the literature, which, isn't actually that big of a surprise. That's why, we like having DEL molecules, because we want to find novel compounds, right? And, when we look at, well, what is the hit rate? When we say, hey, we do understand that we have, a potential hit here, what is the likelihood that it is an actual hit? We actually do really well. We have very high precision. 85% of the things we're calling are being correctly called. And when you sort of work that through to the sort of chemotype level, you do see that those numbers hold up. And in fact, on the sensitivity, maybe it improves a little. On the precision, it sort of drops out. But, that just sort of highlights that there are multiple chemotypes being sort of suggested appropriately by the DEL model. So we also have, of course, assay data, and I mentioned, it's around 2,000 results within GSK. It's a bit more diverse in terms of the chemotypes with actives, right? And, when we look at sort of the performance here, we see a similar sensitivity in terms of, sort of being able to translate the DEL data to the off-DNA data. And again, remember, these are mostly lead-up compounds for unrelated, to PDE3A targets, right? And then the other, sort of, really astonishing finding was, well, what we do know, we're pretty highly confident with it. Almost everything that we called to be a hit turns out to actually be a hit. So this really gave us a lot of confidence Well, and again, similar performance across chemotypes, that when we jumped to, sort of the using the DEL to fish for new sort of chemotypes, that we were likely to have a reasonable sort of bit of success. And I just want to sort of highlight that, as the goal is basically to add more numbers to this row, right? So where do you go fishing? There's a lot of options, right? And I'm just going to draw a line down the screen here, but I took this from a chemistry day presentation one of my computational colleagues made, and it just sort of sizes up, where you can go looking when you start to apply predictions from machine learning models to compounds. And so, here we have, of course, the GSK collection, the DEL collections, a couple logs bigger than that. And these are things that, we have basically gigantic files on our HPC, that of things that we can buy, right? Or that, someone can make in a few steps. And so there's a lot of details here. I'm just going to sort of, for brevity, jump through it. But I basically took the DEL model and generated predictions for 13.5 billion compounds, right? Of which only 0.2% had a score greater than 1, right? So that's a adjusted, in-domain adjusted, odds ratio, right? So that's actually a really low hit rate for a virtual screen. But when you look at that many compounds, you're left with, 31 million plus. And sort of, I could give a whole talk on how one goes from 31 million to 245 diverse compounds. But, I'm just going to sort of jump to, this is where the interpretable ML comes in. And we use this sort of concept of important features to really help us parse through this very, very large sort of pile of opportunity to sort of get after, what's, in this case, readily available within GSK, because it was really easy. We had 245 diverse compounds that I could just, send an email about. And they sort of got routed to the assay. And actually, we were able to just add them to extra rows and columns that would have just been replicates of existing compounds in profiling. So we didn't even really cost us anything on reagents, and we just acquired the data, over the course of months, because we weren't in a big hurry. Right, so what's an important feature, right? And this is a very sort of top level sort of explanation where, we're basically looking for, sort of the exciting, chick here. But again, it's a way of looking at all of the features, right? So all of the columns going through a process where the XGBoost and other ensemble methods basically give you a score for how much the column contributes to the model. And you can kind of look at the directionality of that score, and you can use some sort of fancy statistical sort of Monte Carlo type approaches to eventually get you to a much smaller set, in this case of around 2,000 columns that the model confidently thinks are really, sort of the keys to driving that positive prediction, right? And then we can just, simply take those columns, because they correspond to basically atoms and bonds within the molecules, and paint the molecules, the part of the molecule that contains important features, right? And so here's a very well-known sort of pharmacophore for PDE3A. These are all chemical structures, by the way. That I'm showing, so you could draw them and search for them. And then here's, something that actually is also from Kemble, but wasn't actually, doesn't actually have assay data sort of associated with it. So that was something that, one might want to pursue as a potential new chemotype. And so, just again, sort of summarizing sort of the per cluster result on the internal compounds, right? So this was from slide 8, where, the goal was to add as many clusters as we could to this row. This was sort of the process that we went through. And, sort of the final result after kind of getting these worked through over a couple months of assay was, we identified 20 new clusters with one or more compounds with an IC50 for PDE3A greater than P XE50 greater than 6. So, we added 26% to sort of the known positive chemotypes, and with for the cost of essentially using off-the-shelf reagents and the biologists, sort of valuable time and the time on the sequencing to sort of generate these results. So with that, I'd just like to sort of give a sort of little glimpse into the future. And also, I want to point out that the technology that I used is accessible to anyone with an internet connection. There's really, there's a great community. There's a lot of open source tools. I don't have any specialized machine learning training. I like a lot of people, took the Andrew Ng Coursera course. So, there's really not a lot of specialized training that you need to be able to kind of put these tools together. You do need a lot of sort of Dell knowledge, if you will, and the community of people that have that knowledge is ever expanding. This can all be done on a laptop, except for the featurization and predictions on the big compound sets. Like, you either need a lot of patience or you need a fair amount of compute. I mean, I get this done in a couple hours on our HPC setup, but like if you did run it on just kind of an 8-core machine, it would take 2 to 3 months. It would probably overheat. So you might want to think about, can you run that on the cloud? Looking ahead, we've been doing this now for 2-3 years, and we're really, keen to track all the models that we've built for all the programs and see, what are the aspects of models that are kind of working? What are the aspects of models that are, maybe not working so well? Of course, there's a whole lot you can do, and there are companies focused exclusively on this. And I think there's, a lot of opportunity there around using deep neural networks and combining data, from sort of multiple sources. And I think, we're at the sort of infancy of, some more, sort of complex featurizations, right? I mean, there's a lot of stuff that's been around with graph convolution and MPNNs for a number of years now, but we're really starting to learn how to interpret those. And I think it's the key piece to get sort of the interpretability built in, because that's how you can really leverage it in kind of a discovery team to kind of drive decisions, gain knowledge, and accelerate, the whole process. So with that, I'd like to conclude and just sort of thank people for their time. And, there's such a long list of people that have contributed that I figured it was probably a better, safer thing to just, sort of thank all my current colleagues at GSK, as well as sort of former colleagues. A number of them were familiar with this work, having, sort of worked with me in the past.
Great, thanks, Jeff. That was a really great talk, very interesting work. I think there's a lot of interest in the field in applying machine learning to these data sets.